Thank you everybody for joining this very interesting webinar. So a few housekeeping. So this webinar will be recorded. We will be able also to ask questions. We hope that we will be able, BK will be able to answer most of your questions at the end of the presentation. Also, the recording will be posted on the NF2 BioSolutions website if it's possible, because sometimes it depends on the policy of the company of BK, so we'll let you know afterward. And we have captioning we have a live captioner that is going to do the captioning, high quality captioning. So to turn it on, you have to click on that icon CC that you see at the bottom of your window. And then you need to tap on the three little dots, and then you will see the captioning. Also, I would like to announce another webinar that we are going to do in a month at the end of May. So this is the NF2 month. The month of May is dedicated to NF2, so we are going to do a lot of share a lot of knowledge sharing about the research going on NF2, but also about knowledge about the disease itself. So the next webinar after this one will be about the other research that we're sponsoring that is about NF2 gene therapy. We need your support in order to be able to fund and to fund this different research and also to run these webinars. We need your support, so please donate. You can go on our nf2biosolution.org website. You will see in the menu the link to donate, or you can here scan the QR code, and any donation will be appreciated. So now I would like to introduce Kim Bae-Hoon. I call him BK. He's the research director at PRG, and they have a very interesting research going on and trial to tackle NF2. I met BK in South Korea during a rare disease event that PRG ran. It was very interesting to see that relatively they had a lot of focus on NF2, different research with focus on NF2, and this is one of the research that started already a trial. So welcome, BK, and I will let you sharing your slides.
Yeah, okay, so I will share my slide.
Yeah.
Hello, everyone. How are you? This is a minute. Sorry. This is Bae-Hoon Kim. Currently, I'm working as a research director in PRG Science Technology company and adjunct professor in Busan National University, Department of Molecular Biology. Today, I will present PPT titled with avenues from target selection to clinical development of trinimine PRG-N-01 on NF2-related schwannomatosis. I'm really thankful for NF2 BioSolutions for this webinar. Especially, I hope to say thank to Gilles and Rebecca. With their sincere help, I can introduce our NF2 syndrome asset, especially targeting new pathological interpretation. Before present the talk, I will introduce our company and pipeline and modality and how we found the drug target site. It may take some time and shortly touch the ongoing clinical trials on NF2 syndrome. Then I will present the new interpretation for the disease and show preclinical efficacy of PRG-N-01 with the NF2 model mouse. As a last part of presentation, developmental status and future plan of clinical trial. But unfortunately, in public, I cannot show the efficacy data today, but I can, if anybody interested in our clinical trial, you can email me, is shown in the previous slide. like that in the BK728 and PRGs.com. And I will receive your question then, any interest. So our company is in Busan of South Korea, Eastern Asia, surrounded by China and Japan. Busan is the second biggest city in Korea and located on southeast coast. It has a beautiful sandy beach and a lot of good people there. We are founded on September 2017 by Bong Park, professor in Busan National University. And we do research and develop the treatments for rare diseases like Hutchinson-Gilford progeria syndrome, Warner syndrome, and NF2 syndrome, and for neurodegenerative disease like ALS. Until now, we have funded 37 million from the VC and big company in Korea and received $10 million from national grants and 0.5 million from Progeria Research Foundation and Children's Tumor Foundation in USA. We have around 40 people, including outside advisors. Left top photo is taken in annual workshop in Jinju City in Korea last year. In the middle down photo, the person in the center is blue jacket, is our CEO and founder, Bong Park, and just the right side him is me. We have a global network from chemical synthesis to clinical study and site. As clinical partners, we have a Progeria Research Foundation, Children's Tumor Foundation, NF2 BioSolutions, and Cure Science. For more general information about our company, PRG Science Technology, and any contact information for more detailed question, please visit prgs.com or contact me through email. We have formal pipelines, 3 pipelines for 4 disease indications. Progerin is for HGPS and Warner syndrome. Amisodin is for ALS, and the trinumine I'm presenting today is for NF2 syndrome. Progerin is in the clinical phase 2. Trinumine is the phase 1-2 with the patient, and amisodin is in IND submission for phase 1. First, the patient for HGPS and Warner syndrome is in BCH and the UCMC, have been dosed in this year, January, and last year, June, respectively. In case of NF2 syndrome, first patient was dosed in June last year at Asan Medical Center in Seoul, Korea. Beyond the formal pipelines, we are studying rare cancers, anti-aging, other aminopathies for our next pipelines. We are especially focused on rare monogenic diseases through protein-protein interaction platform. Especially mutation-driven unnecessary protein-protein interaction called as a PPI is our study target. And we are screening PPI inhibitor to ameliorate disease phenotype. Our major modality is a small molecule, and also we are using peptide drug for the next pipeline. For better understanding about the mode of action NF2 pipeline trinumine today, we need to take some time to look at what is the protein-protein interaction and the protein complex in human cell. Proteins works for most processes in the cell with a single unit or as a complex. The protein-protein interaction must be non-random and serve a specific purpose for homeostasis. Forming a protein complex It's normally in specific position or orientation called as molecular docking. The docking can permanent or transient. The PPI can be an important topic in many research areas because of these kind of the homeostasis related working, from basic research to drug development. This slide may explain how loss of function or gain of function can be generated by mutation or foreign protein-driven neo-PPI. I use the neo, the word. The neo does not mean just new. I hope to say neo is a new feature that did not exist before. Left down graph describe the non-perturbed network between proteins in normal cell. In this condition, when some mutations happened in the gene or any foreign proteins from pathogens are translated or embedded in the cell, as shown in a red arrow, new PPI can be generated. And it possibly affects cellular homeostasis and induced disease status. To explain more, genetic mutation in a gene can induce disease phenotypes or not. Please look at the red circle and the star on gene A. Two types of proteins translate to A mutant 1, A mut 1, and A mut 2. If you explain first A mut 1, A mut 1 can generate unnecessary binding to protein X. This binding could not affect any symptom. So we can call it is a silent PPI, it's no influence. But A mut 1 can generate toxic effect to the cell. It is a non-acceptable PPI. So in this case, cells are dying for recycling. And it does not induce the disease phenotype. Then now please look at A mut 2 protein bound to the protein Y. This interaction can generating, for example, cell death pathway turning off, developmental pathway turning off. So then making accumulation of the cellular stresses, and then cell can proliferate pathway turning on, etc. So finally, this acceptable PPI can induce a specific disease phenotypes according to the genes mutated. This binding interface between A mut 2 and protein Y could be drug target site, meaning the forced dissociation of the binding could restore from a disease status to normal condition. This slide shows we identify a disease mechanism of rare disease induced by monogenic mutation, and the disease-inducing neo-PPI is discovered as a target site. Using the target site, we screen lead compound to inhibit the neo-PPI. And we generate or acquire model mouse and treat the lead compound to the model mouse. When you see the in vivo efficacy, then we start preclinical study with the lead compound as a formal pipeline. This is how we find and select a formal pipeline. We think this is our endeavor to find the candidate drug through a traditional way of research. We do our best to make from research to patient care possible. So today we have a 3 pipeline, but today I will explain about NF2 syndrome and trinamine. So this is some summary of the developmental status overview for NF2 pipeline, trinamine. So if you see from the bottom, 2020, we got the lead compound called NF1-A001. And in 2022, we started the preclinical study. And 2023, we got ODD from USA, FDA. And in 2024, last year, we started the phase 1 clinical trial as a medical center Seoul. And between that time also, we got also OMPD from the European Medicines Agency. And this year, we will finishing phase 1 clinical trial in Korea by fourth quarter to this year. And we'll start phase 1 clinical trial in USA maybe from third quarter 2025. And also we are now studying also juvenile toxicology with the trinamine. It will finish by next year, first quarter. So that allowing treatment for over 3-year-old people and 18 years. Because our current clinical trial is over 18 for over 18 years old people. And until now, we published the NF2 related paper from 2015 from mesothelioma. So we published 4 research paper and 1 review paper. And we got grant for this special pipeline from the Korea Drug Development Fund, KDDF. It's around 10 million last year. And we supported from Children's Foundation for the MRI scan the cost. And recent publication is published in Neuro-Oncology, Therapeutic Effect of Novel Drug Candidate PRG-N-01 on NF2 syndrome related tumor. So there also, this paper also highlighted by editorial board, especially as a novel beta receptor 1 kinase inhibitor. So I will explain shortly for the disease for the people see in public. So NF2 syndrome is a rare genetic disorder that is primarily characterized by the benign tumor around the nerve. So the cause is the mutation on NF2 gene encoding myelin protein. And the average age of onset is 18 to 24 years. And primary treatment is now surgery, but there is currently no cure. So common clinical finding vestibular schwannoma, non-vestibular, meningioma, and ependymoma. So until now, there is no cure for NF2 syndrome, and ongoing treatments in clinical trial is mostly a proposed drug used for other cancers or NF1, the selumetinib for NF1. So other treatment also, other cancer or some breast cancer. In case of our treatment, PRG-N-01, it is a first in human patient drug and derived from original research specifically on NF2 missing tumor. I will start a new interpretation for NF2 syndrome. So previous our research on mesothelioma, specific physical contact induced cellular signaling pathway, we discovered that the NF2 and alkyl protein complex suppresses MEP kinase and stain, thereby performing tumor suppression function. Then we look at the Schwann cell and speculated whether NF2 could act as the same regulator like in mesothelioma. When NF2 was deleted, tumor suppressed protein p53 and alkyl protein is decreased upon physical contact. Okay. In contrast, we observed the increase of p53 alkyl protein is if by NF2 overexpression in NF2 deficient Hay193 cell. Through ARKIP GST pull down assay, we found that the ARKIP interacts with the NF2 and also can bind the TGF-beta receptor 1 and 2, which are involved in physical contact-induced cellular signaling of Schwann cell. These findings indicate that the reduced expression of the ARKIP due to NF2 deficiency may be a critical factor in tumor formation. To investigate the influence of NF2 on TGF-beta receptor 1 signaling, we performed NF2 deletion or knockdown experiments. When NF2 was deleted or knocked down, we observed the decrease in ARKIP and TGF-beta receptor 2 expression like that, and yeah, and ARKIP is reduced. TGF-beta receptor 2 also reduced. Considering the significance of ARKIP reduction in TGF-beta receptor-mediated carcinogenic pathway, we investigated whether TGF-beta receptor 1 activity is involved in ARKIP reduction in NF2 deficient condition. We observed the strong interaction between TGF-beta receptor 1 and ARKIP. Interestingly, the physical stimulation induced reduction of the ARKIP at p53 was significantly inhibited by TGF-beta receptor 1 kinase inhibitor TW7197. These results demonstrate NF2 deficiency leads to an imbalance in expression between TGF-beta receptor 1 and 2, and the binding between TGF-beta receptor 1 and ARKIP serves as a major cause of ARKIP reduction. TGF-beta receptor 1 kinase inhibitor TW7197 treatment increased ARKIP expression by blocking ARKIP phosphorylation. This condition induces p53 also expression too, and efficiently decreased the cell viability in NF2 deficient HEY193 human cell. However, it also blocked, but the problem is that this treatment block, because this is a kinase inhibitor, TGF-beta, so the TGF-beta signaling, canonical TGF-beta signaling pathway was blocked. Considering potential side effects on the physical development of adolescents and NF2 patients, there is a need for a drug that specifically inhibits the TGF-beta receptor and ARKIP interaction without affecting TGF-beta signaling pathway. Therefore, so finally, we made the 260 derivatives of TW7197 to develop a selective inhibitor of TGF-beta receptor and ARKIP interaction. Through the library screening, we discovered PRG-N-01 unregulated ARKIP expression like that. It does not affect TGF-beta signaling activation. TW block the smart phosphorylation, but our chemical does not. And transcriptional activity by TGF-beta also stimulation is not affected by our chemical treatment, but inhibited by kinase inhibitor. And also, our chemical effectively inhibits cell viability of NF deficient HEY193 cell. So this slide shows the TGF-beta receptor 1 and ARKIP indeed bind directly protein-protein interaction, and each binding was effectively dissociated through the approach of PRG-N-01 on 255th amino acid region of TGF-beta receptor 1. This is AlphaFold simulation. You can see the chemical approach through the 255 amino acid arginine. Our chemical was treated on the NF2 model, post and crack, NF2 flox mouse, and orally for 9 months from 11 weeks old with the drug diet pellet. We performed PET-CT and histology at 9 months old, and spines with the dorsal root ganglion was sampled at 13 months old. The size of DRG was measured on anatomy microscope, and the pathological schwannoma region on DRG, dorsal root ganglion, was measured by H&E staining. We could see the treated mouse orally has less DRG volume there, and the lower number of pathological schwannoma region, as shown in figure ENF. Micropacet is used to study the tumor formation in a whole body wise. Model mouse treated orally for 7 months was scanned in micropacity. The tumor formation in the body was visualized with color. If you look at the 9 months, the pathological mouse, it has a lot of the red signal. Oh, sorry. In the head and the other body side. But especially the red tumor spot in the head part totally disappeared with the dose-dependent manner. Seen here, there's clear brain. And the whole body tumor site are also, reduced with the dose-dependent manner. Yeah. Here we should remind NF2 syndrome is a progressive disease because gene mutation makes the lifelong effect after disease onset. They keep growing. So we performed another mouse experiment for 5 months with 9 months model mouse, which already has developed tumors on dorsal root ganglion, maybe in the brain. As shown in figure B, the left before treatment and panel. You can see the already tumors formed in the one DRG. From and figure B, this is a treatment as 5 more months grow, it's become bigger DRG tumor. So our chemical treatment is looks like inhibited the growth further. So from figure B to E, we found the PRG-N-01 could inhibit growth around 80% efficacy, already developed tumor in dorsal root ganglion. If you look at the before treatment part in here, these two, the blue spot indicates a Schwann cell, and the brown staining indicates a neuronal marker MVP. The pathological mouse shows high number of Schwann cells are proliferating, and the MVP marker is missing. With the IHC experiment, this experiment, PRG-N-01 treatment could restore neuronal marker MVP with a dose-dependent manner, like that, and reduce the Schwann cell proliferation. And also, PRG-N-01 treatment could restore RKB expression in DRG. Blue spots indicate Schwann cell, like a previous slide. And the brown staining indicates the tumor suppressed protein RKB. Very meaningfully, neurons with the restored RKB expression, like that here, this is collapsed already without RKB. Very meaningfully, neurons with the restored RKB expression shows healthy circle neuron shape and the less schwannoma cell between neuron. So compared to vehicle-treated model mouse, they are in here. So the meaning is PRG-N-01 restores RKB and keeps the neurons healthy. This is the mode of action were found until now and published in the Neuro-Oncology. So we found in NF2 wild type condition, NF2 intact condition, NF2 can interact with ALK protein stabilizing ALK and TGF-beta receptor 2. If NF2 is missing condition, ALK protein is degraded, turn on the ERK pathway and make a growing of the Schwann cell, and TGF-beta receptor expression decreased. So it affects also signaling. But if we treated our chemical binding to TGF receptor one side, but not affected the kinase activity, but inhibit the ALK phosphorylation through kinase 1. And then that induces TGF-beta receptor 1 expression is balanced with the TGF-beta receptor 1, and in making canonical TGF-beta signaling is intact. Yeah, that is the last part of the how we interpreted the NF2 syndrome and how we found the chemical. And this is a clinical side, EPICOS side, but today I cannot share the EPICOS data clearly because only the PI, the clinical trial PI can announce to the public is the EPICOS data according to our protocol and the policy. So I can say some, I cannot say the interim data we got already, but I can say one thing otherwise is because recently we changed the protocol to shorten the clinical phase 1-2 trial in Korea to shorten the time. So we amended and increased the dose and shortened the time and increased the patient recruitment too. So that is approved in Korea already. So and soon we are 4 and 6 and 6 and 12 people more is recruited soon. And clinical trial will finish in the end of this year. And our indication is vestibular schwannoma, non-vestibular schwannoma, meningioma, ependymoma, and tumor size measured by MRI scan, before and after treatment, and the functional response, word recognition score, PTA also we're doing too, and we do also quality of life survey too. And another thing is safety outcome. So until now, we treated the certain patient is treated already 11 months up to 50 milligram. Until now, we don't see any adverse and toxicity issue. This is amended protocol, and yeah, and this protocol in Korea, clinical trial is around 16 people will be dosed, and our chemical can daily dosed, and so as I said before, because the mutation-driven DNA mutation-driven diseases or any syndrome can affect whole life. So and especially NF2 is in need the long-term treatment. It's not by strong inhibitor. So we think positive way our clinical trial. So this is development status from 2020 lead optimization. Now we're doing phase 1-2 with the patient. So I will show you another slide for the more detailed future plan. So maybe September, maybe can be a little bit delayed. Our phase 1-2 trial will finish in Korea. And then we move to IIT phase 2 in Korea, but we submitting phase 1-2 for US FDA. Actually, yesterday we submitted the IND, and we waiting the approval. After approval, we will open the clinical site to one site in USA, and this 2 cohort is not one cohort and 6 people will be recruited and will treated in 6 months. Yeah, and then phase 2 possibly can start. You can look at the not the red one first. I will explain red is later. 2000 next year, the first quarter end, we can start the phase 2 in USA. Yeah, if working great, it's maybe we submitting NDA 2027 and approval 2028. And also we are contacting the Dr. Scott Parkin, the MGH, and Dr. Santo Kesari from West Side, the St. John's Clinical Institute, St. John's Hospital. So we are keep contacting with them for the asking for the PI for phase 1, 2 in USA. Yeah, thank you. Yeah.
Thank you very much, BK.
Yep. Any question? Yep.
So we have a few questions.
Yep.
So the first question is from Jeremy. He's asking if there will be clinical trial in Europe, and I know we are in touch
Yes.
regarding this in order to put you in contact with several
Yes.
NF2 center.
Yeah.
Maybe you want to
I
answer
think
to
you
that.
can explain that one.
Yeah. So in the last few weeks, I became in contact with the Manchester team that works on NF2, and also I'm putting him in contact with Professor Calamarides in Paris as well. So
Mm,
these are NF2 center, two big NF2 center in Europe, and so
mm,
they are making the first contacts currently. So hopefully, yes, the plan BK would like to have trial sites in Europe.
mm. Yes.
Second question, I think you talk about it in the slides. The question is, what
Mm.
are the target tumors type that you are studying?
So we actually in clinical trial in human we made the indication vestibular schwannoma, non-vestibular schwannoma, meningioma, and ependymoma. Four indication we got it. Especially in mouse also experiment and human cell line experiment, we saw already we published for NF2 schwannoma, the vestibular schwannoma related paper is published already. And we have already data with the meningioma data with the low range of the treatment is the meningioma is growth slow down very efficiently. And yeah, so until now we have data, but I cannot say clinical data efficacy, but in cell and mouse
Yeah.
experiment-wise, I can say the schwannoma and meningioma is clearly uh, the growth
Yeah.
phenotype was reduced by our chemical treatment.
And also, as we know, it's very common in NF2 patients. Epidemioma is less common, so obviously. Uh,
Yes, also another thing is I can say we did the C14 radiograph, monograph, or radio autograph wise the measurement of our drug, the distribution in the body. So we compare to plasma ratio. If plasma is 1, we could see our chemical is and compared to 1 to 0.3. So that meaning is the around 30% compared to plasma level. We could see our chemical in the CSF. So our chemical is can get into the spinal the CSF. So that is our chemical is can affect the brain side and the spine side. We think the very positive way.
okay, thank you.
Yeah.
Also, Jad is asking if you could share again your email. Maybe you can put the slide
Yes.
where your email appear, so
Yes.
people
Yeah.
can
1 minute.
know. I think it was on your first slide.
Yes.
Yeah. And Saki is asking, do you think it could help improving the hearing for people that already have some hearing loss?
So I cannot so I cannot say the
Sorry,
result
since
of
you
the clinical trial in human, but so yeah, sorry about that. So you can contact me is personally, so we can discuss and can talk to personally, I think.
okay. So
Yeah.
please, if you have a very specific question about some current results, you can contact BK
Yes,
directly.
yes.
It's
Yeah.
just a legal thing that he has to go through. Uh,
Mm-hmm.
um, are you also in contact with the Dutch Center in Linden? That's in Holland. Um, are you
Mm-hmm.
in contact with any clinic in Holland, Europe?
Oh no, I didn't.
Okay. Okay, so maybe
Yeah.
Mark, you can send an email to if you have a specific contact, and if
Mm-hmm.
you would like to put BK in contact with your clinician in your Dutch
Yeah.
center, you can contact BK, and maybe you can
Yes,
introduce
yes.
them to each
Yeah,
other.
yeah.
Um, will the drug dissolve a tumor that already exists or just stop the development? I mean, what did you see, for example, in the mice thing that you can share? Does it dissolve? Does it reduce the size of the tumor, or does
Mm-hmm.
it stop the development?
Yeah. Can you say one more time?
Yeah, your drug is reducing the size of the tumor. Does it make the tumors decrease in size, or does it
Mm-hmm.
just stop
Mm-hmm.
the development of the tumor?
Yes, yes, got it. So actually we did two experiments with the model mouse. One is from oral, the diet wise, orally treated from 11 years old mice, young mice. And we could see the can inhibit the growth from that experiment. And then we grows the model mouse until 9 months. And then we treated our chemical. We look at the several the the tumor is we monitored. So totally we can say that our chemical treatment is already growth tumor is a further grow is can stopping is clear, and then in some tumor is can be reduced. So we think is may happening the growth reduction is can be happen possibly in by our chemical treatment.
Okay. Okay. Solange asked if there is any NF2 research coordination between NF2 BioSolution and CTF. Maybe I can answer regarding NF2 BioSolution. So, as you see,
Mm,
we are in contact with BK, and we are now running this webinar, but I put BK in contact with different clinics so
mm,
he can expand the trial. And also, we
yeah.
had a research that we sponsored in the past for automatic sizing of the volume of the tumor that
Mm,
he could also be using during the trial, something that we sponsored at Yale. And
Yes, yeah, yeah, sure.
so
Yeah.
BK would like to use this research for measuring the size of the tumor during his trial. So yes,
Yes,
there
yes,
is. And
we gonna have another meeting with the Yale group
yes.
to finalizing the IRD to transfer our data to them. So soon we can realize uh, the the what is better
Yeah.
measurement for yeah, yeah.
And as you saw also in the slide, CTF gave a grant to PRG. A
Yes, yes.
question from Michael. Could you please give an indication if the drug may be a better solution to current available treatment? From what you are seeing, from your knowledge about the current treatment, like
Mhm,
brigatinib, Avastin, or surgery. Uh, so what will be the indication for using your drug
mhm.
here?
So that meaning is
For example, the people joining your trial,
mhm.
how did you selected them? Because they could choose doing a surgery or taking another treatment.
Yeah. So for the recruitment patient, we have a detailed the protocol and the policy. We have it, so I don't remember all. So for in the both the cases, just please email me about that. I can respond as soon as possible. I can do.
O-
Yeah, yeah,
Okay.
I think that will be better. So not talking now, so I can search more and I can reply. So the person want to ask that question is can ask a more detailed one, detailed.
Okay.
Yeah,
So Michael, you can
yeah,
email. Okay. And
yeah.
another question from Michael is, do you have an estimation on the cost of the treatment, or currently you don't have this information on the an estimation?
That cost meaning is after approval
The cost of the treatment for the patient, I assume that's what the question.
in clinical trial or
Not in clinical trial, like afterward, because as you know, in some
afterward,
research,
after commercialization, the
yes.
meaning?
For example, in some research for gene therapy, we know that manufacturing AAV virus is very costly, so we can assume that the drug is very expensive afterward. But
Yes, yes, right, right, right. So, but because this, if our chemical is success, is maybe first approve the drug. If so, compared to selumetinib and compared to the size of the patient with the NF1, we can calculate some way, but we can think it's a little bit expensive. But in USA, mostly supported by government. In Korea, also 90% supported, something like that. But to the nation is not supporting much, supported by much the government. Is we can try to distribute a different way. Yeah.
mm, also like, yeah. A question from Florencia. She's asking if you are in contact with some investigator or researcher in Latin America or Spain.
No, no, not yet. So we should do later. Yeah.
So Florencia, if you have also direct contact, you can also email BK and
Yeah. Yeah.
maybe you can be in contact with some NF2 expert in your area.
Mhm.
Um, and let's see. How long will we have? Oh, yeah, we still have time. Yeah, regarding the side effect of the drug, can you expand? That's a question from Tony. Can you expand on that? Because as we know, for other inhibitor that are being tried, like Avastin or brigatinib, or
Yes, yes, yeah, we know that.
we
Yeah.
know that for some people they have to stop quite early. For some people, somehow they can take it for many years, but for some,
So, yeah, as I know, those kind of the kinase inhibitor is especially for the cancer, right? So long treatment, long time treatment is not easy. So, but compared to that, our chemical is not affecting kinase activity. So actually, also we did because our chemical is bind to TGF-beta receptor 1. That means our chemical can bind to kinase another kinase. So we did the kinase panel. So over 400, the kinase protein in human was treated our chemical, and we found the 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 if that the assay is inhibit over 30%, the the is it can be reported can be affecting other kinase, but through our chemical treatment, we don't see over 30% inhibition any of the 400 over 400 kinase activity. So we think very low adverse effect we can expect because not affecting kinase activity. And mostly also reduced kinase activity a little bit, 10%, something like that. In case of the TGF-beta receptor 1, is our chemical bind to them, but 90-80% activity is still intact. So we think adverse effect will be very low, and our chemical, because of that, our chemical can be treated a long time and daily wise, and we can like any drug like the high pressure drug, so daily treated. So that much, I think our chemical will be safe.
Thank you. A question from Danette, maybe related to the indication. It's like, how will patients be selected to participate in US trial when it will be ready? Do you have already an age? I think you say above 18 years old, right?
Yeah, now current trial in Korea is 18 years old over, but now we are conducting the juvenile talks. So if finishing next year first quarter, after that we can recruit the the between over 3 years old patient we can recruit. So, and then we are talking, we submitted the IND yesterday. If IND is accepted in the FDA, we will discuss with the NF2 BioSolution for the clinical site and the patient recruitment. But this time is only six people, but next year in phase two, we are more people will recruit. So that meaning is that we will discuss with the NF2 BioSolution. We'll ask about the patient recruitment.
Okay. Okay. So you are in the process of for trying it on kids, but that will take longer. But there is a chance that kids can will be able to join a trial in the future next year, right?
Yes, yeah, yeah.
So that answers also to the question of Mark.
Mhm.
Um, is it oral? Question from Madeline. Is it oral or IV treatment?
This is oral treatment, daily. Yeah, yeah, yeah. The
Daily.
tablet. This is tablet.
With the tablets, yes.
Yeah.
Okay. When do you think we'll end the trial of phase two?
Phase two, I think I showed. I, I need to move down.
It's
Okay.
question of for Jade from Jade.
Yeah, and yeah, so sorry. So, oops. Sorry about that. Yes. Yes. Can you see that, right? You can
Yeah.
still can see that. So this, if you see the downside and the dark, the black. Oh,
Yes.
so you can
So
see we supposed to finishing phase one, two in USA this year, and by the next yearly, and we will start phase two, 2026, first quarter, and then
for 1 year.
until yeah, 1 year, and then if I have a success, we're gonna submit IND 2027. This is the plan,
So
the
and
future
in
plan.
2 years roughly.
Yeah,
From
and then, but what I'm thinking is another way. Now, I didn't explain of the red spot, right? Red spot case is if we got great result from the Korea and in phase one, two in USA FDA trial this year, maybe we can submit NDA. Is it possible? Another expectation, the submitting NDA. If we got great result from this year result.
a question is, I assume this question is regarding the current trial in South Korea. Can non-Korean join your trial in South Korea?
Already all the patient recruitment is, I think, most of the finished.
Uh,
So also can also that kind of, because I don't know exactly 100% about the information about the patient recruitment. So can you email me about that, or so I can answer to you?
Okay, so that's what I think that was a question of Michael. If you can email BK
Mm.
regarding that. Question from Mary. Can you explain what you spoke about testing safety in children? Uh, and what will be the age limit for trial in the US?
So because the actually Dr. Scott Flo can ask us about doing the juvenile talks. So we are now conducting like this, the juvenile talks, may finishing in the 2027 first quarter. So after that, if this is success, so we can recruit the children
Mm.
from next year, I think.
From age of 3 years old, so 3 years old and above, right?
Yeah, yeah, over 3 years old.
Yes. Okay.
Not below, not the.
Are you aware of contraindication to of you with use of this drug? So if you use another drug, or if you have different condition, do you know if there is some limitation on using your drug, contraindications?
So then mean this thing, right? So this similar question, this one. Can you see that? Do
Yeah,
you
yeah,
see
yeah.
the what? Yeah. So in case of another medication, the taking. So I think the no tumor treatment medicine is allowed together. But if you stop dosing before 3 months, before into our clinical trial, 3 months stop is possible. And so there are unacceptable medication list also there. So if you send the email, I can send that medication list too. But usually normal medications such as high pressure, diabetes, etc. are not allowed.
Okay. Uh, do you assume that this drug will be taken all the life of a patient? That's your
Yeah,
assumption.
yeah, yeah. So the lifelong wise treatment we are thinking about.
Yeah. Some questions about also access for the trial for the drug. So some Saki is asking for India, someone else from Philippines. Are you in contact also in Asia for
No, no, no.
no?
Because the we need to do uh, but we will do one by one, I think. Yeah.
Okay.
Mm.
Okay, so for all the people that ask questions about local trials, if you have the contacts in your area, in your clinics, um,
Mm.
you want to introduce BK to your contact, don't hesitate to send him an email about it.
Yep, yep.
It's exactly
Definitely.
what I'm doing with the contact I have in UK and in
Yep,
France.
yep, okay. Yep, yep.
Yes. Um, uh, how Um, question from Saki, how effective is this in comparison to brigatinib? I don't know if you say you cannot really share
Yeah, but
publicly some
I
early results.
actually in the cell data and the human patient cell data, we published to compare with the brigatinib and selumetinib. Actually, the uh, this paper, we put the data in there. So effective wise is similar with brigatinib with our chemical, but toxicity wise, our chemical was better.
Okay. Um, Someone asked, what if a patient doesn't have any symptom, but I assume he has tumors, so he could be a candidate as well, I assume, right? As long as the tumor are growing. I don't know. Are you asking for active
Mhm.
tumor, meaning history of growth of the tumor before they can join the trial?
So I think the mandatory is for the MRI
Yeah,
scan, I think.
yeah. Several
So
MRI for the
MRI
last few
scan.
years. So you are looking at several MRIs to see the growth or just the last one?
Yeah, and the comment from the clinician about the MRI scan is needed to join the clinical trial.
Yeah.
Yeah.
Okay. About the US trial, as you said in your slide, that it will be at most likely at MGH and in St. John's in Los Angeles, right?
Yeah,
Uh,
we think now that way. So yeah, we think yeah, the Intuit 2 is a Scott Plugin. Dr. Scott Plugin is the PI, right? Intuit 2.
yes.
But that is
Okay.
the not formal the clinical trial, but we can open to there later. We are discussing about that. And for the west side, actually for phase 1, 2 is soon we will start. There is we are contacting Santos Kesari. And then other clinical site is we are looking at too.
Okay. Okay, so thank you very much. I think there is a few more questions that we can share with you.
Yep.
And
Eh,
if you answer to them, then Rebecca can make sure that the person that asked the questions will get the response.
Yeah, yeah, sure. Okay, okay.
Yes. So thank you so much, BK.
Yeah,
And
yeah,
hopefully we will see you again once you have data that you can share about your trial
sure,
in
sure.
South Korea and
Okay.
in US, and we'll continue to work
Yep,
together to find more trial
yep, yep.
sites. Well,
Okay, okay. So I will, I will contact you if
thank
we
you.
have any progress. I will contact you.
Thank you. Thank you so
Yep.
much. Thank you,
Thank you
everybody,
so much.
for
Yeah, I appreciate about this opportunity to introduce our asset to the patient and family and public. Yeah, thank you so much.
thank you, BK. Have a good
Yeah,
day.
bye bye.
Good night.
Yeah,
Bye-bye.
thank you. Bye
Bye.
bye.